Okay, so tonight I'm really excited to have one of our very own SNARL postdocs here tonight. He's actually been working here so far a very short time this summer. He just got up here, but he was here all of last summer studying the Yosemite toads up in the east side of Yosemite. And Dave Daversa is a postdoc from UCLA, and his research examines how wildlife movement influences disease risk. In collaboration with the National Park Service and the Kretz Center for California Conservation Science at UCLA, he's working to better understand the movement and infection risk in endangered Yosemite toads with the goal of developing data-driven reintroduction strategies to strengthen their vulnerable populations. Dave received his Bachelor of Science degree in forestry at Virginia Tech in 2006 and completed his PhD in 2016 at the University of Cambridge. And since then, he's done postdoctoral research at the University of Liverpool and the National Great Rivers Research and Education Center in Alton, Illinois. And some of you may know Dave from his running. He's a frequent runner out at the Whitmore track. So I'm sure some of you have seen Dave running out there. And I'm sure Dave is really going to be excited to be back here again at SNARL, getting over the Whitmore track. I'll be over at the Whitmore pool in the morning. So summer feels like it's getting to be in full swing here for us at SNARL. So it's really great. So I'm looking forward to Dave talking with us about his work with the Yosemite toads. His seminar is titled Down with Disease, Managing Pathogen Threats in Yosemite Toad Populations. So Dave, you can take it away.
Right. Thank you, Carol. Thanks for the introduction and for inviting me here to give the seminar today. And thanks to everyone for spending this evening to come and listen to me talk. I am just going to share my screen real quickly here. All right. So yeah, once again, my name is Dave Daversa. I am based at UCLA doing a postdoc there. I guess I'd like to start by acknowledging, maybe a few of you noticed, it's kind of cheesy, but I included the name of a Fish song in my title. For those of you that didn't notice, there's a somewhat popular music group called Fish out there, and they have a song called Down with Disease. And I intentionally put it at the beginning of my title in hopes of maybe attracting some of the folks from Mammoth to this talk, but I'm not sure if I was so successful. But Down with Disease, the title of this song, it does pretty much sum up what our human society has been going through in the past couple of years with the COVID pandemic. Hopefully we're rounding a final corner with it all, but you know, humans are not the only species out there suffering from a pandemic. In fact, the amphibians that I've been studying have been facing their own global pandemic for the past 2 decades. All right, and this pandemic stems not from a virus, but actually a little microscopic fungus known as the Kitrid fungus. Okay, the scientific name of this fungus is Batrachochytrium dendrobatitis. So a lot of the folks in the field, like myself, that study this parasite just refer to it as BD. For this talk, I will try to refer to this parasite as Chytrid, but if I say BD, I apologize, and please just know that I refer to the Kitrid fungus, okay? This Kitrid fungus is waterborne, so it thrives in the water bodies that amphibians like to breed in, okay? And it only infects amphibians, so humans don't have to worry about it. However, it infects pretty much all species of amphibians, and because of that, it's known as this really extreme case of a generalist pathogen, okay? And the way it works is, here, I'm gonna show you a little video quick of Chytrid kind of under a microscope. Okay, so it has 2 life stages, okay? The first, the infectious stage, are these little tiny spores that swim around in water bodies, okay? And they are actually responsible for infecting amphibian skin cells. I should mention that this is a skin infection that amphibians get, all right? So these little spores, like this video shows here, they swim around, and they kind of latch onto the skin of amphibians and they colonize it. And if they are successful in colonizing it, they will get inside their skin cells and they'll develop into what are called sporangia, which are shown. These are like the fruiting bodies in that video in the center parts. And they are actually reproductive bodies. So within those sporangia grow more spores, okay? And there's more spores that keep getting produced until those sacs that you see comprising the sporangia, they will burst and release more spores out into the water, potentially swimming on to infect other animals, or perhaps coming back and reinfecting the same animal from which it came. All right, and this little microscopic fungus has really been wreaking havoc on amphibians all over the world. Okay, so dating back to the late 1990s, there's been scientific paper after scientific paper that has come out with headlines such as this, documenting these mass mortalities associated with the arrival of the chytrid fungus into these populations. And one grim example of this comes actually from the Sierra Nevada here in California, where SNARL is. All right, and I actually first started working with chytrid here in the Eastern Sierra, where I am currently based. And at the time, I was working with a group of researchers from Berkeley and UC Santa Barbara, tracking a wave of chytrid outbreaks throughout the Sierra Nevada in not toads, which is what I'm going to talk about today, but a high altitude frog called mountain yellow-legged frogs, which the middle picture here, you can see it. Okay, so I first got exposed to this fungus as it was decimating these poor little mountain yellow-legged frogs in the Eastern Sierra, and I got really interested in this topic, and so I got some opportunities to actually move to Europe and study how the same parasite was wreaking havoc on amphibian populations there. Okay, so in particular, I spent a couple of years in Spain. So there's a map of Spain in the center part of the screen there, and the little dot just north of Madrid is where I was working. There's a chain of mountains just north of Madrid called the Sierra de Guadarrama, and up in these mountains were amphibians that were being affected by this chytrid fungus. Okay, and while I was there, I got really interested in how the movement patterns of amphibians, such as their seasonal migrations to and from the ponds that they breed in, like the one shown in the top left here, how their migrations were impacting their infection risk. All right, and even not just migrations, also just, you know, the way they were moving from pond to pond or moving around on their daily basis. How that was impacting their infections. Okay, so I went and I put little tiny radio transmitters on common toads, like those. These are common toads in the bottom left here. They're actually breeding, but I put little transmitters on them so I could track their migrations. Okay, and then after that, while I was doing that, I started noticing the species of newt that you see in the top right here. They're called alpine newts. And I became really fascinated about how they were moving around among these little alpine ponds, like the ones shown here, when they were breeding. All right. And so both of these studies that I did, both with the toad migrations and these breeding movements with alpine newts, I kind of did with the hypothesis that, okay, these animals are moving, and because of that, they're going to be these kind of vectors of chytrid. They're going to be spreading chytrid around at these sites. All right. And I actually found the opposite. All right. I found that, you know, whether it was migrating from ponds as toads did, or just moving, hopping from pond to pond as they're breeding, as alpine newts did, these movements between water bodies actually help them recover or remove their chytrid infections. And so I kind of scratched my head at this a bit and was wondering what was going on. So actually a large part of my PhD work, I brought these alpine newts into the laboratory and studied their movements in very high detail. Okay, so this is a video of some behavioral experiments I did with alpine newts, where I put them in tanks and I gave them the choice to move freely between water and terrestrial habitat, and the water had different levels of chytrid fungus in it. Okay, so this is kind of what the video looked like. I did this for a period of a week, and this actually showed that when animals contracted chytrid infections, and as the infections were proliferating on them, they actually got out of the water and they spent a lot more time on land. Okay, and I did a follow-up experiment that showed that when they are actually on land, it actually is not good for chytrid. Chytrid doesn't like when animals are in these upland terrestrial habitats. And so it kind of actually cleared their infections. And it was this cool little kind of behavioral mechanism that at least these newts employ to keep their infections in check. All right. And it may be one reason why, unlike other species like the mountain yellow-legged frogs, we never really saw loss of mortality from chytrid in these alpine newts. All right, so that's just a summary of my previous research. And I got all this great experience in Europe, and I brought it back with me to the US to do this postdoc at UCLA. All right, and I just want to talk a little bit about the institute's kind of sponsoring this postdoctoral work. It's kind of a partnership between the Kretz Center for California Conservation Science, which is a center at UCLA that is all about conserving the biodiversity of species in California in particular. Okay, so they do this by sponsoring research like my own focused on threatened and endangered species, but also they have a research element looking at urban ecology, obviously with an emphasis on the wildlife of Los Angeles. They also have some projects involving how animals are adapting to ongoing droughts in the Western United States. And there's also a strong genetic component. So there's doing a lot, the folks there are doing a lot of whole genome sequencing for the purposes of wildlife conservation. Okay, and this Kretz Center is kind of embedded in this broader institute called the Institute of Environments and Sustainability. And I'm not trying to like praise like UCLA, but I will say after having been there at the IOES for a year and a half, it's a pretty amazing institution because it's very diverse. It brings together faculty from a lot of different disciplines to tackle the common issue on how to build sustainable environment under the current climate. All right, so we have folks like myself, ecologists, going out to the field and doing very basic science research. But the office next to mine, there is an environmental economist, and the office down the hall for me, there's a sociologist, and there's politicians there. So it's a really cool, diverse group of people trying to work together to address common problems relating to the environment and sustainability. All right. So this fellowship that I'm working under, the Kretz Fellowship, it likes to promote partnerships with non-academic agencies, also promoting California conservation. So I reached out to the National Park Service, Yosemite National Park in particular, and I had a colleague there, Rob Grasso, who is shown here doing his thing up in Yosemite. He is the aquatic ecologist of Yosemite National Park. And we started talking a couple years ago about some of his efforts to develop conservation strategies for threatened Yosemite toads. All right, so that's what I decided to do my postdoc work on, and that's what I'm going to tell you a bit more about now. All right, so Yosemite toads, they are endemic to the Sierra Nevada. They only occur there. All right, and they occur in these high elevation meadows that Yosemite and surrounding areas of the Sierra are very well known for. Okay, so this is one of the sites that I worked at called Gaylor Meadow. All right, it's in the Tioga Pass area. I'll show you a map here in a bit. So you can see it's these kind of high elevation grasslands. Okay, and toads, they are these classic pond breeding amphibians that for a large part of the year you don't really see them. They're kind of hidden in burrows on land. Okay, but once a year in the spring, right around this time as the snow is starting to melt up in the mountains, the animals migrate from their terrestrial refugia and they kind of flock to these wetlands like the ones you see here. And one thing I didn't realize until I got out there and started working with them, unlike the newts and the other toads I had been studying in Europe, these toads actually don't like lakes. So actually in Gaylor Meadow, you can almost see in the background there's a large permanent lake there that it would be just perfect for breeding in. But toads, for some reason, they don't like deep permanent water bodies. They prefer these very shallow little boggy, almost like puddles that are scattered throughout the wetland areas here. After the snow starts melting. All right, so here's a photo from that same meadow, just kind of zoomed into one of the breeding areas of toads. And you can see they only breed for about 1 to 2 weeks, but when they do breed, they just go crazy. So in this small little area, you can see there's 1, 2, 3, 4, 5, at least 6 pairs of animals doing their thing. All right, and so I guess the other aspect of Yosemite toads that is relevant to this talk is that they have different life stages. Okay, and so I'll go through their different stages of development and their life cycle here in a bit. All right, and I just want to highlight, I included this photo down in the bottom right. This is the first Yosemite toad that I ever saw. It was last year in mid-May, right around this time. A colleague and I went out to Gaylor Meadow, this meadow here, to kind of check it out. And in mid-May last year, it was still fully covered in snow. So we got out there and we concluded that, okay, still too early. The toads aren't here yet. And we're going to come back in a week or two and see how things are then. All right. And we're walking back. And what do we see as we're hiking out? But a female Yosemite toad slowly crawling across the snow. So I guess she was extra excited to get the breeding season underway. All right, and so there is a bit of information on kind of the history of these toads in Yosemite. So there were some folks that did surveys of toads dating back to the early 70s in the same area up around Tioga Pass of Yosemite. And so they would go to these meadows every year and they would capture as many animals as they could. Okay, and they kind of looked at kind of these population trends throughout the 70s. And the notable thing about these data are in getting around the late 70s, 76, 77, particularly in male toads, you see this massive decline, sudden decline in their numbers. Okay, and ever since that time, their population sizes have kind of dwindled and they never have really bounced back to the sizes that they once were. All right, and so for this reason, Yosemite National Park is really interested in efforts to conserve these animals and not just kind of conserve existing populations, but also kind of bolster their populations, even increase their population sizes through strategies such as reintroducing animals that they breed in captivity. All right, but before they do that, before they start releasing these animals into the park, it would be nice to know what caused these declines in the first place. And it's still an open question. They don't really know, but there is some evidence that this Kitrid fungus did play a role. Okay, so there was a research group from San Francisco State University that a few years ago, they actually went to museums and they looked for preserved Yosemite toad specimens in museum collections. Okay, and they were actually able to find a lot, even some specimens dating back to the early 1900s. Okay, so they got some starting in early 1900 and from every decade up to 2010. All right, and so they developed this method that they were able to actually detect or sample for chytrid from these museum specimens, and they developed kind of a time series of chytrid in Yosemite toads from these preserved specimens. And I guess the main thing to note here is that in the early 1900s, not seeing much chytrid, but oh, look at this. In the late 1970s, they start seeing BD positive samples from the Yosemite toad museum specimens right around that same time that we start seeing their population declines. Okay, so this is not direct evidence that chytrid was the culprit, but it's at least some loose evidence. It's suggestive that chytrid may have played a role. All right, so my whole motivation was to come out to these sites up in Yosemite and really do some thorough systematic chytrid surveillance in these Yosemite toads, because really that hadn't been done before. So I really wanted to get a grasp on how prevalent chytrid was in these populations. All right, so this is just a map of the areas that I went to last year. This all happened last year in 2021, okay? And so for folks out there that are not familiar with Yosemite National Park, it's in northeastern California in the Sierra Nevada, and this is a map in the top left of the boundary of the park, okay? And the elevation increases pretty sharply as you go from west to east. So I focus more in high elevation areas in the Eastern Sierra, right around what's called Tioga Pass. Okay, so if you look at the zoomed in map, you can see the main road going through Yosemite here, Route 120 or Tioga Pass Road. And I had 5 sites in the Tioga Pass area. Delaney Meadow, Lower Gaylor Meadow, that's the meadow that I showed you pictures of a few slides back. Dana Meadow, which you can actually drive by if you're going through Yosemite, and then a bit further afield were Spillway and Mono Pass. Okay, and I visited these sites once or twice, 1 to 2 times a week throughout the entire summer. Okay, and I'll point out that I was fortunate enough to not have to do this by myself. There was a really ambitious and keen undergraduate student at UCLA, Molly Posta, who's shown here. She came up and did an internship with me for the summer. So I was fortunate enough to have her help. She's actually just probably preparing for her final. She's a 3rd-year marine biology student at UCLA. But anyway, she came up last summer and helped me. And the 2 of us, what we did is like I said, 1 to 2 times, every 1 to 2 weeks, we would go out to these meadows and we would capture as many toads as we could, okay? And we would sample them for chytrid infections. And the way you do that is really easy to explain to everyone now, thanks to COVID, all right? It's very similar. We capture toads and we take swabs and we don't stick it up their noses, but instead, remember that chytrid is a skin infection, all right? It produces a skin infection. So we actually rub their bellies, all right? We just want to get a tissue sample from their skin. So we swab their bellies, okay? And we go back and just like they do with your COVID test, we do PCR now. We run it through a PCR and see if we can detect any chytrid DNA on those swab samples. All right. And I just want to give a shout out to SNARL, because all the PCR processing did happen here at SNARL. And a special thanks to Alexa Lindauer and John Imperato for processing the majority of the samples for me. And like I said, as I promised, I would tell you a bit more about the different life stages of toads, okay? And we went out and collected swabs of all their different life stages, okay? So a bit of basic background in amphibian biology. You probably all are familiar with this, but so females will, if they successfully mate and reproduce, they will lay clutches, what we call clutches of eggs in these water bodies that they breed in, okay? And clutches eggs, they look like a string of pearls, okay? And these eggs, they develop, and eventually they'll hatch out into tadpoles, okay? This is a silhouette of a Yosemite toad tadpole here. And those tadpoles, they'll kind of grow and develop for not too long, about 4 to 6 weeks. And at that point, it's time to metamorphose, and the tadpoles turn into these little tiny toads. Okay, I call them metamorphs because they are recently metamorphosed. Some people call them toadlets, and they're small enough to stick on your fingernail. Okay, just to give you a perspective. Actually, this picture here is of Molly measuring a little metamorph, and you can see this tiny little dot here. That's the toad. Okay, so they're tiny little animals, and if they're lucky enough to survive and go into hibernation, they'll come out the next year in a different life stage, what we call subadults. Okay, they're basically juveniles at this point, and they will spend 1 to 2 years growing, developing as juveniles until they become they become sexually mature and they start reproducing as full-blown adults. Okay, this is an image here of a female toad. All right, and so when we would go out to these meadows, we captured as many adults as subadults that we could every time that we went out, and we swabbed them. Okay, then when the tadpoles emerged, we captured 30 tadpoles from every site, and then we got 30 swabs from tadpoles. Okay, one time, because they weren't around that long. Okay, so 30 swabs every site from tadpoles, and then once those tadpoles metamorphosed, we got swabs from the little metamorphs as soon as we saw them. So right after they metamorphosed, we captured 50 of them at every site, swabbed them, gave them a little break. Around that time, it was July. I'm going to show you actually like a timeline of all this work on the next slide. The metamorphosis happened around mid-July. So we swabbed them then, we gave them a break. We went back a month later in August, caught another 50, swabbed them at every site, and then gave them another break, and then returned in September as it was starting to get cold and these animals were about to hibernate, and we got one more round of swabs. Okay, so all that field work and molecular analysis provided kind of this swab data set that gives like a nice temporal profile of chytrid infections throughout the season. All right, so I'll just quickly reiterate. The blue bars are adult male toads. The yellow bars are adult female toads. Okay, so we caught as many as we could every time we went out and surveyed these meadows. And the main thing to notice is that we were pretty successful in capturing adults in the early stages of the season, because this is when they're out breeding and there's loads of them. All right, so they're easy to find, but like I said, they're only breeding for a couple weeks. So after they're done, they kind of leave the water bodies and they disperse, and they like to hide in mammal burrows. So it's really hard to find them, which is why you see we kind of capture one or two males and females here and there, but we didn't really get much of them in the later stages of the season. But the subadults, on the other hand, we caught as many as those as we could every time too, and we had a lot more success. Okay, sometimes here, in June, 50 animals that week, that's pretty good. And it was a lot more consistent. There was a decline towards the end, but still a lot more consistent data from those animals. All right. The tadpoles, like I said, we went out and we collected swabs from them once within that 4 to 6 week time frame that they were present. And then finally, the little metamorphs that metamorphosed from being tadpoles in mid to late July. We swabbed them at 3 different time points. When they first came out in mid-July, which, it didn't happen at the same time in every site. So that's why we have swabs across 3 different weeks here. But this is still what we consider the first time point. Then a month later in August, then 1 month later in September. Okay, looks like we got 1,600 swabs total. All right, and what did the swabs show us? Well, first of all, the main point is that there were BD positive swabs that we collected, which means that chytrid is circulating in Yosemite toad populations. All right, so we can dive a little deeper into the data and look at how the prevalence of chytrid infections varied across these different life stages that we sampled. Okay, so this figure shows the prevalence of BD or chytrid prevalence in the samples that we collected, or prevalence is just the proportion of samples that we collected that came back positive for chytrid. All right, first thing to note is that none of the tadpoles tested BD positive, and we actually weren't quite surprised by this. There were some folks that went and swabbed tadpoles in previous years, and they've never found BD positive samples from tadpoles. So it's something about tadpoles, Yosemite toad tadpoles, not all tadpoles, but Yosemite toad tadpoles, that they're just not that susceptible to infection. All right, but once they metamorphose, there's a little bit of chytrid starting to pop up. Okay, these little metamorphs, the ones that fit on your fingernail. Not a super high prevalence, on average about 15% prevalence of chytrid in the animals that we sampled. But man, these subadults, these ones that were metamorphs, you can consider them 2020 metamorphs, the metamorphs from the previous year that had hibernated. When we went out and swabbed them, they had super high rates of infection. Okay, so on average, what is that? 50% of the samples we collected from subadults came back BD positive. All right, a lot more than any of the other life stages, including adults, even though they're a lot bigger. All right, so the adults, both males and females, were kind of in the middle of the road. All right, and so we can take these infected animals, these infected samples, and we can actually quantify the severity of infection. We call this the infection load, and it's essentially the amount of chytrid DNA that we detect from our swabs. Okay, so you can think about it in terms of COVID. If you get swabbed from COVID and you have a very strong infection, there's going to be a lot more viral DNA detected on your swab than from someone who they may be infected, but they have a weak infection. So you'll both get positive tests, but your test, because you have the stronger infection, will actually have more DNA on it. All right, and so we can use that as a measure of infection strength or infection load. And when we look at among the infected individuals, again, the subadults, much stronger infection loads, although the females, there was a lot of variation, but they exhibited high infection, stronger infections as well. The metamorphs in the males, for whatever reason, the infections that they did contract were relatively weak. All right, so, but this pattern in the subadults, I want to dig into this a little more. So we will kind of look at how these infections varied throughout the course of the season. Okay, so these two plots here just show, on the top, it's the prevalence, like I said, the proportion of samples that we collected that came back positive. And in the bottom here, we kind of break down the prevalence by the total number of samples that we collected, which are shown by the bars. And of those samples, the number that were chytrid positive, which is shown by the black line here. All right. And the first thing to note here is that there's a much higher prevalence of infection in these subadults at the very beginning of the season. All right, and then we will see that the prevalence gradually declines as we go throughout the season. Don't pay much attention to this ostensible spike in infections at the end, because that's based, if we look down here, that's based on like 1 or 2 samples. So we don't have much confidence in it. The shading here denotes the confidence intervals, which is all based on how many samples you collect. So, this is a very wide confidence interval. We don't have much confidence in that. So we can kind of, it kind of suggests that there is a steady decline in chytrid infections in subadults as the season progresses. I'll show you the same thing for the adults, but I'm not going to dig into it much because, like I said, we weren't able to get a good temporal profile of swab data from the adults. I guess the main thing to take away from the adult data is that they are competent hosts of chytrid. They do get infected. They do sustain infections. Okay, so that might be relevant. And then that leaves us with these little tiny metamorphs here. What's going on with them? Just to remind you that the sample design was a bit different. So we swabbed them when they first metamorphosed in July, 1 month later in August, and then right before they started hibernating as it was getting cold in September. All right. And when we look at these data, there's some really cool stuff, some really interesting stuff happening. All right. So in July, there's a very, very low prevalence of chytrid infections in these new metamorphs. Okay. Hardly any of them are infected when they first metamorphose from being tadpoles. But every time we go back with each subsequent month, there's kind of chytrid kind of proliferating in them for whatever reason. And this is kind of interesting. Never super high levels. You can see in September, it was on average about 25% of the samples that we collected came back positive. But still, there's this interesting increase in infections in them. All right, we can also break that down across the 5 sites that we looked at, and it does vary a bit, but it is interesting to note that all except for 1 site in July completely BD negative. So none of the animals at these sites metamorphosed and were infected. Nevertheless, some of them did contract infections throughout the season, and that's kind of interesting. There's a bit of variation here. Spillway is one of the higher elevation meadows, and for whatever reason, it kind of bucks the trend. But, in general, there's this gradual increase in infections in metamorphs throughout the season. We can also look at the infection levels of those BD positive samples and metamorphs, and it follows the same general patterns. So these small metamorphs are developing infections later in the season, and those infections that they do develop are getting stronger throughout the season. All right. So this is kind of interesting. We can start to put some pieces together here, and there's a kind of an interesting story that emerges that I'm thinking a lot about. First, if we focus on the left screen, this is the metamorph data. So like I said, they come out, they begin uninfected largely, but they start to develop infections throughout the season, okay? And then we can start to think about those subadults that we collected in the very beginning of the season. And these remember were the metamorphs of 2020 of the previous year, all right? We can kind of put this together in almost this hypothetical calendar. And it raises the question as to, okay, gradual rise in infection in metamorphs, and then there's this long period of hibernation that we know nothing about. And then when we go back and we capture animals again that have overwintered, these subadults, there's a huge spike in their infections. All right. And so this is, it's kind of touches on a topic that is interesting to the field, the niche field of chytrid ecologists. And something I've been thinking a lot about is what happens to chytrid infections when these animals are hibernating? And no one really knows, but here is some evidence that it's actually doing something to promote the proliferation of chytrid infection in these animals. All right, so just some key points to take away from these data. Like I said, chytrid is present in Yosemite toads, all right, and it's mostly affecting the younger life stages like the subadults, all right, and these results are kind of providing support for the evidence that I started uncovering during my work back in Europe, that okay, much of what is known about chytrid comes from the breeding phases of amphibians, because that's when they're in the water where chytrid thrives. That's when we can find them the most. And you would think, okay, chytrid thrives, it's aquatic. You would think most of the infection happens when they're breeding in water bodies, but it turns out that a lot of what's happening terrestrially outside of the breeding season, such as when they're hibernating, may be important to understanding infection risk in these animals. All right. And so these are some key points to take away from this first field season. And of course, sorry, I'll go back. The work raises more questions than answers, as always. All right. So for example, some questions I'm thinking about are, how do infections develop in those yearling metamorphs that they metamorphose and none of them are infected, but by the end of the season, roughly 25% of them have developed infections. What is the reason for that? Don't really know. Would like to explore. Again, what happens infection wise during this long period of hibernation? Okay, is there something? Are they aggregating? Is their immune system really compromised that they come back the following season and they're a lot more heavily infected? And finally, it still remains known what is the actual cost of chytrid infections in these animals. There have been some experiments done by some of my colleagues in those little metamorphs. They did some infection experiments and they gave them very high doses of chytrid experimentally, and it did prove lethal. Okay, so at very high doses, chytrid is lethal, but we're not quite sure yet how that translates into natural populations in the field. All right, so just real quickly, I'm taking this first year of work on Yosemite toads, and I am back now at SNARL for another round. I'm doing some follow-up work to kind of build on that data set I got last year, and really I'm doing much more targeted surveys. I'm going back out and I'm looking specifically for last year's metamorphs. So this year they will be subadults because they did go through hibernation. I'm going to go back to the same spots in those same meadows and look for these animals and get more swabs of them to see if this pattern holds, that after hibernation, chytrid prevalence is a lot higher than before hibernation. All right. Also, like I said, the hibernation question, I've been wanting to dive deeper into this question for a while. So I'm currently seeking funding and resources that I might be able to do some experiments where I actually simulate hibernations in the laboratory, because, you know, up in Yosemite, these animals are buried under snow. It's a bit difficult to observe them and sample them in the wild, but experiments like those I did in Europe can be very valuable to answer such questions. Okay, so that is one area of research for myself going forward. And finally, going back to that work I did in Europe, I do think there is a value in, you know, tracking the movements of these animals to get a sense of their behavior and how their movement patterns and different behaviors are influencing the patterns of infection that we have seen. And I would just like to conclude by discussing another topic that we're all very familiar with. You know, I came to study these disease in these toads, but we cannot ignore the influence of climate change. All right, and I've been fortunate enough to live in different areas of the world, and in other areas that I have lived, climate change is discussed and largely acknowledged. But in California, climate change is really felt. You know, you can feel it here intimately. You can feel the smoke from wildfires, and you can feel the crackling of this desiccated vegetation up in these alpine meadows that are normally nice and moist. Okay, and this is concerning for us, and it's very concerning for animals like these Yosemite toads that rely on shallow water bodies to reproduce. All right, and this is just a picture I took. These are Yosemite toad tadpoles here in one of the breeding water bodies in the meadows, and you can see it's very shallow. And sure enough, I went back the following week and it was all dried up before those tadpoles were able to metamorphose. All right, so climate change, you know, we can't ignore it here. And having moved to California and confronted it directly, it's inspired me to kind of bring in this topic into my work. I actually avoided it for a while. It's kind of a bandwagon in my field, and I didn't want to follow the trend. But now that I'm here in California, it's forced me to swallow my pride and tackle the issue. So I am actually currently working with other UCLA faculty to map out the different pathways that climate change and infectious diseases like chytrid actually interact to affect the survival and overall viability of wildlife species. Okay, so I won't go into the details, but it's our hope that the framework that we're developing, like the one shown here, can inspire research that kind of goes out and really quantifies the combined impacts of climate and disease stress on our California biodiversity. And with that, I will finish. I can't finish without thanking all the folks that are supporting me. Once again, Yosemite National Park, UCLA. I'd also like to thank SNARL. They are very gracious in hosting me. I stay at SNARL when I'm doing my field work. That's where I am now. Carol and her family has really welcomed me and been really great supports. And also the Mountain Lakes Research Group. They've been doing amphibian disease research up in the Sierra Nevada for the past 2 decades. And they've really been supportive of this new Yosemite toad work, and they've lended me a hand in various ways in getting this project off the ground. I'll just finally note that my contact info is up on the top right here. I'm happy to discuss this stuff more if you're interested in it. You have questions that you don't get to ask now. Feel free to email me. I'm happy to discuss this stuff anytime. And I know there may be some folks listening actually at SNARL now. I'm happy to meet in person while we're all here and chat about this stuff more, just feel free to reach out. And with that, I would like to thank everyone again, and I will address some of your questions. So Carol, I think you gave them instructions to put them in the Q&A. So, and if anyone, feel free, please, to speak up as well and chime in with any questions you may have. Thanks.
All right, thanks, Dave. And I am, as Dave mentioned, for those of you out there with questions, you can either type them in the Q&A box, or you can type them in the chat, or you can just raise your hand with the little raise hand button at the bottom, and we'll let you talk. So while people are sort of thinking about their questions, I've got tons of questions for you, Dave. So just while the first question's coming in, one thing I was curious about is obviously there's a lot of work here at SNARL over the years with the mountain yellow-legged frogs and chytrid and similar endangered status for that species. And a lot of the early work with Roland Knapp showed that certainly chytrid is a serious factor, but also sort of predation by the introduced trout as a factor in their declines. And would you say for the toads that chytrid is, like, how important is chytrid in their sort of endangered status versus things like other things that you mentioned were habitat, maybe shrinking habitat, water, things like that?
Yeah, that's a good question. So, I mean, with any endangered species, one main issue is determining what are their threats, so that could be chytrid, diseases like chytrid, it could be predators, it could be habitat loss. So yeah, the mountain yellow-legged frogs, chytrid is a clear limiting factor on their populations. Yosemite toads, it's tough to say. We're not seeing mass mortalities like we do in the mountain yellow-legged frogs. So although chytrid is in these Yosemite toad populations, they may just be persisting with it. It's almost like they might get a cold, but they're not dying from it. Personally, out there, my observations, I feel like climate is a major, the ongoing droughts are a major issue for toads because of their preferences for where they breed. They breed in these very shallow areas. They like water that's not more than 2 inches deep. All right. And so, when there's a drought, that's not good because those areas are going to dry out a lot faster than normal, sometimes faster than the tadpoles can develop and get out of there. So I think drought is a major issue, and that's, and kind of in conjunction, my prediction would be that stress from droughts will make them more susceptible to chytrid infections, and perhaps more susceptible to dying from infections. I have no way of, I can't look at that in my data yet, but it is a topic of interest that I would like to look into further.
Cool. Okay, anyone out there? John, I think I saw your hand up. John, do you still have a question? I can let you talk if you want.
Well, I'll try.
There you go. We hear you, John.
Yeah, you were following a similar vein. We've had some of their earlier studies were showing mainly the frogs that got the Kitrid fungus. Some of them, a lot of them died. Some of them seemed to be immune to it. Some of them got infected and their skin got bad and they survived. And the deal there on the planted trout was the tadpoles used to be able to repopulate. And when they planted trout in some of these remote mountain areas, they couldn't do that anymore. And I'm wondering too, you're saying the Yosemite toads breed in shallow water, but did they always do that? Or is that the survivors that are existing now? Maybe there was portions that bred in deeper water, had the tadpoles that all got eaten. I'm just looking at a lot of these from former lectures, and we have had droughts before, I think, in bristlecombs. another lecture. What was about 720 years ago? They had a doozy in these in the tree rings, it shows. Anyway, that's about it. Just other things to do in your spare time.
All right. Yeah, that's a very interesting comment. I mean, there is literature like dating back to the middle of the 20th century on Yosemite toads, and they've always found them in these meadows. But yeah, in longer time scales, I can't rule out the possibility that they bred in lakes. Because they're not currently found in lakes, it does rule out the trout carol as one of their threats. So the trout are not in these shallow little puddles. But yeah, I've never really thought about that. Maybe there was a time where they did. Yeah, that's an interesting point.
And it looks like Dave Scott Cooper's got a question in the Q&A box.
Yeah, I can read it off
Yeah.
here. Across amphibian species and from your own work, it appears that chytrid impacts on individuals depend on the amount of time amphibians spend in aquatic versus terrestrial habitats. Generally, aquatic species and stages are more vulnerable to infection. Do you have any hypotheses about why Yosemite toads appear to be different? Yeah, that's a good question. When I read this question, I think of the tadpoles. You would think tadpoles they are fully aquatic. They're always in the water, and they should then be more prone to chytrid infections, but yet we see no infections in tadpoles. We don't really know for sure, but I have discussed this with Rob and other people working on amphibians up here. And one thing is that because the water bodies are so shallow, they tend to warm up in the summertime, and chytrid doesn't persist above 30 degrees Celsius. Okay, and I was taking the water temperature of their breeding water bodies last year, and by late June, early July, some of these water bodies were getting up to 30 degrees, so that might allow tadpoles to escape infection. Also, tadpoles, they develop really quickly. They're only there for like 4 weeks, so it may just not be long enough for them. Finally, one aspect of chytrid biology I failed to mention is that it actually infects keratinized portions of amphibian skin, and tadpoles only have keratin on their mouth parts when they're developing. When they metamorphose, the keratin spreads throughout the rest of their body, but as tadpoles, it's only on their mouth parts. Okay, so there's just not as much surface area for toads to get infected. With that said, it's the same as mountain yellow-legged frogs only have keratin on their mouth parts, yet the tadpoles carry very high infection loads. Yeah, and it is a good point about aquatic versus terrestrial environments, and it's one I actually expected to see the opposite trend in the metamorphs. If you'll recall, the metamorphs infections gradually developed in them as the season progressed, and I expected the opposite. Because you can imagine when they first metamorphose, they have just gone through a very taxing endeavor of metamorphosis, and their energy levels are sapped. So you would think they're less able to fight off pathogens like chytrid. And also I noticed that they were gradually moving away from water bodies throughout the season. So they were becoming more terrestrial, and by September, they were largely terrestrial. Yet it was in September where we saw the highest prevalence of chytrid in them. So it is a good question. I think the terrestrial habitat does play a role, but there is some nuance there that warrants some further examination.
All right, Dave, I've got one more question for you, if there's no more, because I really want to know what's going on in hibernation, and
yeah, you and
what
me
are
both.
they doing? Where are they? And have you thought about getting up there at that time of year? Obviously, there's a lot of snow, but if it's possible to sort of sample during that time.
Yeah, I'm not much of a Nordic skier. Maybe I should get some do some skiing lessons so I could get out to these sites. I suspect that what's happening, like I said, when these animals like to hide in mammal burrows, like squirrel burrows up there. So I'm guessing they go deep down in these mammal burrows and they hibernate there. And one hypothesis, I guess you would say, that I have, is that they aggregate in little clumps in there in the wintertime. And it's that aggregation that allows them to transmit infections. And when they come out the following season, a lot more infected because of that hibernation behavior. It could be, I mean, you like keep in mind when animals, these amphibians are hibernating, they're almost dead. Their metabolism is down to about 0. So there's no real like immune defense happening, you know, there's no real kind of basic maintenance going on to keep these animals healthy. So, you know, they may get exposed to chytrid when they're active, and then go into hibernation, and just that lack of basic function may allow chytrid to proliferate. But I suspect it's something in their aggregation behavior, and it's something that I would love to test in the future. Getting out there in the wild, I don't know, Carol. There's a
I bet
lot
we
of
have a bunch of
snow.
recruits on this call.
Yeah. Hey, if
some
anyone's
ideas.
interested in doing that, I would love to chat with you. And if you have any ideas for how you could do that, I'd love to hear them. Yeah, because you can imagine, I mean, there's a lot of mammal bros out in those meadows. And so it's really a needle in a haystack issue. But if someone wants to help me try, I'd be happy to entertain it.
All right, well, I'm game. I think, John, did you have one more?
I'm just going to turn the light on real quick.
If John did. All right, well, thank you again, Dave. Really great having you here. And thanks everyone for attending. And this talk and all the other ones are recorded, so you can find them on our YouTube channel.